So these are the two sets of axons approaching the base of the limb, and the upper dorsal region of the limb contains one of these repellents. It's called ephrin B, but the name doesn't really matter. What's important is that it will repel motor axons and growth cones that express the receptor EPHB, whereas ventrally there's a different repellent, ephrin A, which is capable of binding to its EPHA receptor. So in work that Arthur Carnier and Ed Laufer have performed, this repellent system, which is mirror symmetric, ensures the growth of these axons into the right halves of the limb. So let's see how this happens. So initially, axons are growing along, they reach the base of the limb, and initially the red axons will encounter the dorsal mesenchyme, the dorsal cells in the limb. As a consequence, the green axons will encounter the ventral source of ephrin A signaling. This is a productive engagement that leads to the collapse of the growth cone in just the same way that we saw. And so no matter how many times the red axon tries to innervate the dorsal half of the limb, that growth cone will be collapsed and it won't be able to grow any further. If by chance the red axon samples the ventral half of the limb and the green axon the dorsal, then that productive receptor engagement doesn't occur, there's no collapse, and so the axons are free to pursue this path in a perfectly unimpaired way. So in this way, this repellent signaling system ensures the initial pattern of motor innervation of the limb.